My name is Todd Durham, the Senior Vice President of Clinical and Outcomes Research at the Foundation Fighting Blindness. I'd like to welcome our two speakers today, Dr. Christine Kay and Mr. Robert Rapoza. And this session was originally titled Medical Ethics and Trial Participation. Yesterday, one of our speakers, Ken Goodman from the University of Miami, who is an ethicist, attended. Unfortunately, today he's not available. So I've invited Christine and Rob to cover any number of areas from their field of expertise. So I'd like to welcome you to this session. This session will last approximately 1 hour, and we're going to leave plenty of time for your questions. I'd like to introduce the two speakers first now, and then I'll let them give their prepared remarks. Christine Kay is a vitreoretinal surgeon. She joined Vitreoretinal Associates in Gainesville, Florida in the fall of 2014. Prior to this, she was an assistant professor and director of the Retinal Fellowship and Retinal Genetics Service at the University of Florida. She graduated magna cum laude from Harvard University with a degree in neuroscience and went to medical school at the University of Florida. She completed her ophthalmology residency at the University of South Florida, where she was chief resident, and completed her vitreoretinal fellowship training at the University of Iowa. During her fellowship at the University of Iowa, she developed a particular interest in inherited retinal disease, and this has become her niche in her retina career. Dr. Kay is very interested in characterizing natural history of disease in patients with various inherited retinal diseases. And she is the director of electrophysiology, retinal genetics, and clinical trials now at Vitreoretinal Associates, involved as principal investigator in 15 IRD clinical trials. She has a very large IRD patient population with over 900 patients in her clinical database, and with 600 of these patients genotyped. She's very interested in genetic testing and has written articles and given lectures on the importance and logistics of genetic testing for IRDs. She organized a faculty of international experts in inherited retinal disease and has taught an instructional course on retinal genetics and gene therapy at the Academy of Ophthalmology annual meeting for the past 7 years. Our second speaker is Rob Rapoza. His career spans more than 24 years of preclinical and clinical research in both ophthalmic and cardiovascular indications within academia, discovery and development of pharmaceuticals and novel medical devices, and CRO settings. He brings extensive experience managing phase 1 to 3 ophthalmic clinical trials, both within the U.S. and internationally, with therapeutic experience in glaucoma, AMD, DME, RP, MACTEL, and achromatopsia. Mr. Rapoza primarily oversees AURA's U.S.-based clinical operations for those trials involving anterior-posterior segment ocular indications, and he's been with AURA since 2017. All right, I would like to turn the floor over to Christine first.
Okay, welcome everyone. Is this on? Can you hear me? Yes. Okay, good. No. Okay, now it's on. I can hear it. It's a much bigger room than what we used yesterday, so it's a little bit less intimate, so I'm going to scoot up here. And I wish that Dr. Goodman was still here with us, so if you didn't get a chance to meet him, he's an ethics professor with a very deep and soothing voice, like a fireside chat. So we had a really interesting talk yesterday about the ethics, and he asked some really interesting questions about what would you do? You know, would you treat a patient, do a trial with a patient? I just lost my voice. But anyways, I don't have the deep voice. So what I'm going to focus on today is a little bit of the history of ethics and what we do in clinical trials as clinical trial investigators. I'm going to talk a little bit about my experience as a principal investigator, which means the physician that's sort of running the clinical trial at their clinical trial site. I'm going to talk a little bit about the conversations we have, and probably the most important conversation that I have is the informed consent conversation that we have with either the family member who's going to be in the trial or the pediatric patient and the entire family. And we'll talk a little bit about some of the things that we even do training on as clinical trial investigators in order to understand some of the ethical principles behind clinical trials and being able to execute a clinical trial in a just and fair and correct manner. So let's just think back to sort of where all of this began, and it began before this. You can trace some of this back to some of the, and Dr. Goodman was telling us yesterday, even unfortunately some of the Nazi history and trials being done and things being done in an unethical manner. So we've learned a lot from that internationally, and certainly nationally we've had some very significant mistakes in history as well. The Tuskegee trials, which were in the 1930s, were unfortunate, very unfortunate part of history where unknowingly African-American male patients who happened to have syphilis were being studied in a trial unbeknownst to them and being not treated, intentionally not treated, and the purpose was to learn about the natural history of a very severe disease called syphilis. And there were so many things wrong with that trial that I can't even begin to go through how many ethical principles were at play there. But we've learned quite a bit about this as a country and internationally as well. The Belmont principle was one of the important things that came out of that. And so these are not things that I was mentioning yesterday. We don't inherently know this in medical school. So actually, even in medical school, we take classes on ethics. And then certainly when we become principal investigators, and even as physicians, we are invited and requested to complete courses on ethics, HIPAA, which you probably have heard about when you go see your physician. You sign forms on privacy. And then we as investigators take multiple courses on ideas of ethics within a clinical trial. So what does that mean? So in a clinical trial, we actually do care that we explain risks and benefits to patients, that we are executing the trial in a just and fair manner, that we aren't excluding populations, different populations of race or of age. Importantly, with that conversation, there are vulnerable populations, and that's one of the key words we think about in ethics of clinical trials, that there are vulnerable populations. So what does that mean? So that one of the most common that I see would be a pediatric patient. And so that, in the U.S. is anybody under the age of 18. They can't legally sign their own informed consent form in a clinical trial. We do actually have them sign something called an assent or assent form. And so there actually is a separate form for most trials that the pediatric patient is requested to sign. And then the parent, who's over 18, of course, needs to, or the legal guardian needs to sign the official informed consent for a trial. And so then we think about principles such as benevolence, so basically doing the right thing and making sure that things are in a positive favor of the patient. We take that into the equation for risk-benefit calculation, so we need to think about safety and adverse events and the risk the patient is going to go through, even risks that you don't necessarily think of. The obvious risk would be any damage or injury to the organ being investigated, any damage to the eye. But then there are other risks, there's risks including privacy violation. So how are we careful with documentation? How are we careful with protecting the patient's privacy and rights? There are confusing ethical considerations with siblings in a trial. So what if there are twins or siblings and there's a randomization that's going to occur where one sibling is going to be randomized potentially to treatment and the other sibling might be randomized to placebo? Is that fair? Is that ethical? How do we have those conversations with families? There may or may not be correct answers to all these questions, but we at least we communicate with the families, and we have these difficult conversations with families. Trying to explain a placebo-controlled trial to any patient or family is always a challenge. Of course, the patient would prefer to be the one randomized to receive the treatment. I'd say a very average number of the way that we randomize is 50-50. So there are trials with different numbers, but often the FDA or the sponsor is going to design a protocol to be randomized. 50% of the patients are going to receive treatment and 50% are going to receive an oral, if this is an oral drug trial, a placebo. If it's an injection trial, if we're going to be injecting in the eye, how do we do that? We can't, inject water into the eye. That's not safe. You can't take a patient. What if it's a subretinal gene therapy surgery? We can't take a patient to the operating room and put them to sleep and pretend to do a surgery and not do one. That's not ethical. And so for gene therapy trials, the ethics become really interesting because we need to think about ways to randomize or have a sham or a placebo control, so that the idea there is that we're not going to have patients who have a placebo effect, because the FDA is worried about that. But then we have to, on our side of the clinical trial, manage that ethically. The way that the FDA typically gets around that for subretinal injection trials is doing a dose randomization. So there's 2 different doses, a slightly higher and a slightly lower dose, and potentially even the company knows is sort of the correct dose, and there are ethical implications of whether this is the correct path forward. And then we often have a third arm of getting control. So of course the patient knows they didn't go to surgery, they're control, and they just go through an observation period with Luxturna, which was approved several years in 2017. They had a deferred treatment arm. So those patients who were in that trial waited for 1 year, and then at 12 months, they were able to have their treatment, which was subretinal injection of Veretagine Naparvavac, which has now been FDA approved. So all of these considerations are at play when we're talking about clinical trials and designing clinical trials. These concepts of privacy, of beneficence, of justice, of equality, of protection of the patient's rights. These are all very important things that we, at least every year, sometimes every 6 months, I have another online certification from either a sponsor or from within my own clinic to recertify and retrain myself, to remind myself, and I take a little test online and have to pass it and send my certificate and put it into my regulatory binder for all of the companies and trials that I'm involved in. So these, at least I think it's important to say that we have learned something as a country, and hopefully internationally as well, from things like Tuskegee and horrible disasters previously to that. And I think that we now integrate ethics into our clinical trials in the way that we execute these clinical trials. And so that, I think, is something that is actually very enjoyable to be a part of, and to be able to say to my patients, that I have looked through the scientific validity of a product, and I've done my research as a PI to understand the scientific validity and the scientific rationale. We spend it depends on the patient, the trial, and what we're explaining, anywhere from 30 minutes to an hour plus on the informed consent conversation. Sometimes I have a phone call the day before to save some time from a busy day of screening. To give a little idea of what a patient, and I will up in a minute, we'll of course hear from our other speaker and our questions, but just to give an idea of when a patient's going to come in for one of our inherited retinal disease trials, they're often getting themselves into a full day of screening. These protocols, and in order to get through the inclusion-exclusion criteria, and then to get through the entire protocol of screening, that often can take a day, sometimes even 2 days. Some of our tests, visual fields, ERGs, FST, these tests can take an hour plus, and then we have to do the other eye, so these are time-consuming days for these patients. So sometimes having a phone call with the patient and the family to have a little heads up of what you're about to experience before we get even into the room to have the informed consent conversation, that might happen. And then the day of screening, I'm going to spend that first half hour, an hour, talking with the family, kind of prepping them, doing the informed consent, doing my initial exam, and then the patient's going to spend a day with my clinical coordinators going through an extensive amount of testing. And this is just the screening visit, so then if it's an oral drug trial, then we're going to randomize them and put them on an oral drug and see them every month, every 6 months, depending on what the trial protocol is. If it's an injection trial, we have trials where we do injections, and I forgot to mention, we do sham injections, so that's interesting. We actually, in order to have a placebo arm, we lean a patient back, we literally, this is dictated by the protocol, I'm not making this up, but we literally pretend to do an injection on the patient's eye with a blunt syringe, and they know that they've been randomized to sham or treatment. They know the concept, they don't know which one they've been randomized to. And that's the idea that the FDA requires randomization and being blinded. We don't like to use the word blinded anymore in ophthalmology trials, but a patient not knowing, so being randomized and not knowing what therapy. And then I, as the investigator, I'm always the masked, we call it masked, not blinded, investigator, so I don't know what therapy that patient has had, and I just do their exam, and then one of my 3 other doctors in the clinic is my sub-PI or sub-investigator, and they are unmasked, so they obviously know if they're injecting real drug or if they're just putting a blunt syringe to an eye. So I have multiple trials like that going on, and patients go through the trial, usually every month in injection trials. And then thirdly and lastly, we have these subretinal gene therapy trials. I always do the surgery for those, and again, there's usually a common scenario would be dose randomization, so dose A and dose B, or the patient doesn't go to surgery and they're just followed as control. So that's just a very overview of what we're thinking about with ethics and all of the sort of logistics of what happens with clinical trials, and I'll stop there.
Thank you, Christine. Hello, everyone. My name is Robert Rapoza. I'm the Director of Clinical Operations for an organization called Aura Clinical. We are a CRO, or a clinical research organization. What we do is we provide services really to whomever in the research industry needs it, whether they're an individual physician trying to work on development of their own discovered drug or medical device, all the way through small or very large pharmaceutical companies or medical device companies. We offer a wide range of activities or services, really, all the way from just simply management of the study across a variety of different sites, to also including data management, statistics, and advice from a regulatory perspective with various regulatory bodies. So before we get into too much discussion about clinical trials, I'd like to see a show of hands of those who have participated in a clinical trial thus far. And excellent, thank you. And then a show of hands for those who will be or are thinking about participating in a clinical trial. Excellent, thank you. So what I'd like to do first is, as we're discussing sort of safeguards and patient safety and data integrity, which of course are really the key points that we want to maintain through the entire clinical trial activity and duration of a clinical trial, I wanted to sort of take a step back. Before we even get to a clinical trial, there's a lot of preclinical, non-clinical, or discovery work that needs to occur. And we're talking about years of effort, millions of dollars, and essentially, if you look through the literature, what we tend to see as far as metrics on that is for every 5,000 or so drugs that are investigated non-clinically, or again, before it gets to the clinic, really only one of those gets into the clinic. So a lot of research, a lot of work goes into this. And even to get that one entity into the clinic, not only does it involve discovery, it involves a lot of animal research, and as well as validation of different processes that will be utilized both preclinically and clinically to ascertain the data that we need to, again, demonstrate both that the drug is safe as well as there's efficacy, or again, that we are able to show that the drug works. So one of the things that as patients you really need to think about is self-education. There's a lot of resources out there available to you. Whether you go onto the FDA website and really look at the resources that they have to investigate what it means to be a clinical trial, what are the safeguards that are inherent to the way that we do clinical trials today. Really, it behooves you to really dive down into that level. There are other resources available to you. For instance, there's a website called clinicaltrials.gov, for those of you who aren't familiar with that yet. It is a website that, for the most part, within the United States, or really any FDA-approved clinical research that's ongoing, really needs to be posted online and available to the public. And within that, it's going to tell you a very high-level summary of what the protocol looks like, what's entailed, and then really what's helpful for you as patients is it's going to list all of the clinical sites that are involved in that clinical trial. So you can see, is there a physician near me who is participating in this particular clinical trial? It's also searchable, so you can search for a specific medical indication. So if you have RP, you can go into clinicaltrials.gov, search for all the RP trials. You can see not only the RP trials that are ongoing today, but those that have occurred. And if particular clinical trials have occurred and they've ended, you can also go in and see some of the data. So it's a way of just really creating a good baseline foundation for yourself to educate yourself on what clinical trials mean. So one of those aspects of clinical trials that you really want to consider when you're thinking about a clinical trial is what is the study phase. I'm sure you've all heard, it's a phase 1 trial, it's a phase 2, it's a phase 3. They do have different meanings. So a phase 1 trial is often referred to as the first-in-man study or first-in-human study. And this is an opportunity for the drug, or again, the medical device, I don't want to exclude medical devices, but for the investigational product to be tested within humans, just so we can ascertain that it's safe. And then it goes to a phase 2. And again, we're looking at safety there. We're looking at a larger population. In the phase 1, we may have actually tested the drug from a safety perspective in a healthy population. Not always, but that is a possibility. It may be within the population that is actually experiencing the disease state. In phase 2, it is going to be tested within the disease state population. And we will, at that point, really start hoping to see some sort of signaling within the data relative to efficacy, or again, how well the drug is working in its intended response. And once we get through that phase, then we're going to go to the phase 3, and that's really the pivotal trial. That's going to be the very large trial. So whereas in the phase 1, we would enroll, somewhere between 20 to 100 study subjects, the phase 2, maybe a little bit more, somewhere between 50 to maybe 1,000 study subjects. The phase 3 could be quite large. So we're typically talking several hundred to several thousands, or sometimes even several tens of thousands. So it's a very large study, and it's pivotal in the sense that not only do you need to demonstrate safety and efficacy, but there may even be longer term implications of the study research or the data that would be collected. So study data collection doesn't end the day that you're done with the trial. Obviously, all the patients who are involved in a trial need to undergo all of the assessments and participate their full duration. But the expectation, too, from a safety perspective for investigators is still to hear from their patients. If 2 years after you've completed a trial, suddenly you start having other symptoms, you are expected to have that discussion. And is it related to the clinical trial? We don't know. But why it's important to collect that data is not only do we look at things from a per patient perspective, but we have to look at it from an aggregate perspective as well. So for instance, if we have adverse events, if suddenly we start having, or a patient comes in and has a cold, let's say, just a cold. Well, that's meaningless into itself. It's an unfortunate circumstance, but again, into itself, it doesn't mean anything. But if we start seeing 25% of the population that within a day of having received their injection, that they're having a specific symptom, then it does reach a new level of significance. So I've briefly talked about sort of this assurance of safety. And I've mentioned the FDA. So the FDA really is, from a clinical perspective, the first level of review. So a protocol needs to be submitted to the FDA, needs to be reviewed. Quite often there would be it would be submitted to what we call an IND, or investigational new drug. And it's an application to the FDA. Quite often that would be preceded by a pre-IND meeting. So that's an opportunity for an investigator or a pharmaceutical company to sit down with the FDA and say, this is the drug that we're intending to develop. This is the planned clinical process that we intend to get it from where we are to hopefully market. And the FDA has an opportunity to provide feedback. So ultimately, what we need to do is not only address being able to demonstrate our endpoints, but meet the expectations of the FDA. So along with the FDA is the Institutional Review Board, or the IRB. That's another set of eyes that are on the documentation. The IRB's role is really all about patient safety. They want to look at the protocol. First of all, they will have an opportunity to have their questions addressed. Then they're going to review it not only from safety, but they also want to look at it from a sense of redundancy. Has this work been done already in another form? So they need to look at it from a number of different perspectives, and they come back with a ruling relative to whether you can proceed. Quite often there are questions and there's back and forth, but eventually what you're working towards is IRB approval. A part of the IRB approval is also working with them for the informed consent form. Christine already addressed the informed consent form to some degree, whether it's an assent for a pediatric population, or just the informed consent for the adult population. Let's see, who am I missing as far as safeguards? Oh, yes, this is an important one. So generally within study protocols, we have incorporated what's called a data safety monitoring board. You'll see this definitely incorporated into a study protocol, definitely, within really the earlier phase studies, the phase 1, the phase 2. However, regardless of what phase the study is in, the expectation or the inherent nature of research calls for the ability of a DSMB to be pulled or convened at any time if there is a signal suggestive of a safety concern. So it's an opportunity for key opinion leaders within a specific medical indication to take a look at the data, where it stands today. If there is any sort of safety concern, it's an opportunity for them to review the data in detail and make a go, no-go decision. Or alternately to say, you know what, we don't have enough information to make a go, no-go decision. So we need more information and further investigation needs to happen. So there's a lot of layers, there's a lot of regulations. I didn't even go into good clinical practices or the Declaration of Helsinki or ICH. Again, you can access all of that information on the FDA website. I would invite everybody here to do so. But there are medical professionals, such as Christine, as well as your own physicians. If they're involved in clinical trials, they've been trained and they have the information that they can share with you, again, to provide you the necessary information so that you're informed in the decisions that you're making relative to participating in clinical research. And with that, I'll open up the floor, I believe.
Yeah, we would love to have questions if you have any. We have 2 mic runners in the back, so just raise your hand and they'll find you.
Thank you. My question is, Robert, you just said that clinicaltrials.gov is for FDA-approved sites, and it was my understanding that it is not, and that often there are stem cell and unaccredited, un-FDA-approved trials on clinicaltrials.gov, and so patients need to exercise due diligence when they're looking at it. Now, perhaps that's been remedied in recent years. I know the FFB was involved in a major effort to try to get it remedied, and I would appreciate an update on that. Thank you.
I can't address that specifically. I'm not aware that any FDA unapproved clinical trial was ever allowed to be posted on clinicaltrials.gov. So if there's anybody with other information to that, please share.
Yeah, hi, this is Todd Durham. I think I can answer. There are clinical trials that are listed there. In fact, there's one particular field on the clinicaltrials.gov where it says, is this trial under FDA oversight, yes or no? So we advise members of our community to check that one in particular. And there have been stem cell researchers who've not had any FDA oversight, and we caution folks against those.
And given that we live in the state of Florida, I'll tell you another bad story, which is the Florida story. I won't say specifics, but there were some stem cell trials which were not under IND, so investigational new drug applications, probably not under FDA watch, and these were some trials that happened a few years ago where patients were invited to spend $20,000, which is a huge red flag. Usually, almost exclusively in a clinical trial, you should never be asked to pay for anything. You should be covered your expenses, even travel expenses, sometimes even a small stipend for your time, but certainly any expenses related to the trial should be covered typically by the sponsor. So that's always a red flag if you're asked to pay for something. And so this was unfortunately a trial where patients were asked to pay $20,000, and then they had stem cells, I believe, extracted from their skin, so the idea of a fibroblast, and spin it and do something with it. And then they were either injected intravitreally into the retrobulbar space. The intravitreal was unfortunately concerning because several patients had bilateral injections and then developed bilateral retinal detachments with proliferative vitreoretinopathy, or PVR, which means the eye scarred itself up internally and died with a bad retinal detachment. So these patients went aggressively bilaterally blind within a few weeks. My partner, who's one of my retina partners here at my practice, was I don't know if he was the chief resident or fellow at Bascom Palmer, so obviously this would be the place where these poor patients end up trying to get repaired, and he had the experience and he'll tell you this conversationally, of trying to repair one of those retinal detachments. He was, of course, not involved in the original trial or treatment. He was trying to save this patient's eye, as a chief resident at Bascom Palmer at the time, and these eyes end up needing silicone oil and still failed and weren't able to see. So bad things can happen, terrible, terrible outcomes can happen. That was a very not well executed, not ethically executed, and not good to enroll in trial, but that's an example of you do need to take caution. I still have patients, even now, will find things on Google or find things on various websites and send me an email and say, is this a good idea, this trial in maybe another country, and I always say, let me look very carefully at that for you before you sign up for something. So you should really have whoever your physician is taking care of you, your ophthalmologist, hopefully a retina specialist, or specifically an IRD specialist who's well versed in these trials, should really just take that extra step to make sure that they approve of whatever you're interested in thinking about prior to even contacting those sites to hear more. But you really, clinicaltrials.gov is helpful. It doesn't, as we say, exclude any potential bad thing from sneaking in. Having an NCT number, having an IND number, you can even ask. And then knowing the sponsor, researching the sponsor. Typically sponsors will have a website. You can read about their scientific advisory board, their medical advisory board, their team. You want to know, being a reputable company is an important thing, and having some prior data. And then probably most importantly, you want to know your site, know the investigator site, and know that investigator and their history, numbers of trials they've been involved with, et cetera. So word of mouth is helpful in communication, but definitely be very careful because there are still, unfortunately, some things out there that are not safe.
Good morning. I'm Eric Bernardo. I'm the president of the Foundation Fighting Blindness Pittsburgh chapter. Thank you for your presentation, and here's my question. I have X-linked RP with the RPGR gene defect, which is being very heavily researched and has a lot of current clinical trials associated with it, some that are even in the phase 2-3 stage of the trial. The reason why I haven't jumped into any trials myself is because I happen to be monocular. I only have usable vision in one eye, so I'm really not at the top of anybody's list to be in any trials for safety and safety reasons, since I only have one eye to really lose. And that kind of brings me to my question is, with your knowledge and expertise in the clinical trial space, what is the timetable between where a lot of the late stage RPGR trials are and actually being approved by the FDA?
So it is a very actively researched field right now. I was involved as an investigator in the Biogen Nightstar trial, which you probably are aware, unfortunately released their data relatively recently that they didn't quite meet the primary endpoint of the static perimetry, 7 decibels over 5 grid points, and now the FDA requires those are pre-selected and repeatable with statistical significance twice. So very hard endpoint, so it doesn't really mean that it didn't work. It just didn't work enough to show FDA approval. I treated several of those patients, and I firmly believe that we saw benefit. And I'm now an investigator for Mira GTX/Janssen. We are currently enrolling for the trial that you mentioned, the phase 2/3, I think it's what it's labeled right now, which is bilateral treatment. So we do require with the inclusion criteria for that particular trial that both eyes meet criteria and both eyes can be treated. AGTC, who's here at this event and very visible at this event, has several RPGR trials going on, Skyline and Vista, and they're still in the treating unilaterally phase. However, typically, and please correct me someone if I'm wrong, they typically still require that both eyes meet certain inclusion and exclusion criteria. And so typically, and it makes sense ethically for a clinical trial, we don't, for a treatment or interventional trial, we do typically exclude someone who's monocular because of the potential risk and the unknown safety. Until a drug is FDA or a product is FDA approved, we can't say, and even we can't ever really say anything for certainty, but until you have FDA approval, you can't really speak to the safety and efficacy of a product to the extent that you would want to for a monocular patient where you don't have a spare eye. And so there is potentially the option of natural history trials for you. I'm also a site for the Mira GTX is doing, sorry, the 4D, I'm getting my trials confused, 4D Therapeutics, which is the intravitreal attempt to repair our PGR gene. They are doing an interventional trial as well as a natural history trial. I can't recall the inclusion criteria if they would allow a monocular patient in the natural history trial, but at least it makes sense that that would be, a plausible trial. Of course, that's not too exciting a trial for many patients to be in a natural history observational trial. However, it's critical to the field. I don't know if anybody here was involved in RUSH-2A, the Foundation Fighting Blindness USH-2A trial. Very critical example of how a natural history trial can pave the way to refine outcome measures and help the FDA understand endpoints in that disease and even open doors for ProQR Therapeutics treatment trial for that disease. So natural history trials are critical. Timeline-wise, it's always longer than we want, but if something's in a phase 2/3 or a phase 3 and they're pivotal, they could be within a couple years to an FDA approval if things are working. Now remember this, a couple caveats. What is the goal enrollment? So what's the total number? I heard the term, some trials have 10,000 patients. That's never happening here in our world here of rare orphan diseases. My trials typically target enrollment for a phase 1/2 is like 45, 40 patients, 20 patients, 15 patients. Even when we get into our phase 3s, it might be 80 patients, 100 patients max. Or even in some of the more rare diseases. So first of all, you have to enroll all those patients. Usually 12 months is a common outcome endpoint. So we got to get everybody enrolled and get everybody to their primary 1-year endpoint. And once we have all that data, then there's a data cut, and then things can get submitted to the FDA for the review, for the end of phase, so EOP, end of phase meeting with the FDA. We have this regulatory meeting with the FDA and submit your data. And then look at the history of Luxturna. So that was supposed to be a 6 months expedited approval. Most drugs, I think they have a year for the FDA to get back to you and give you an approval, yes or no. I remember speaking at the FDA advisory committee just as a speaker in October of 2017. We flew up to DC and got to see what the FDA looks like, and it actually did not take 6 months to get Luxturna's approval. I think it was 3 months later, it was December 2017, so they had a super expedited approval because the data looked really good, safety and efficacy-wise. So things can happen really quickly. I think Luxturna is a good example of a 3-month approval from the moment they had that meeting with the advisory committee to the moment of approval.
Thank you. Good morning. My name is Sandy Matthews. I have a question. It's a little off topic, but I haven't been able to figure out where to ask it. I get, like many other people with inherited retinal diseases, articles from friends and family about different things they've just heard. And one I got recently was on a drug that's got FDA approval for treating alcoholism, Antabuse. Has anybody heard anything
This
about
must be
that?
everywhere on the news right now, because I can't tell you how many patients email me about anti-abuse, or disulfiram is what the drug is.
Okay.
So I honestly haven't looked into it enough, even with my patients having emailed me about it. But so what I do know about it is there some preclinical potential efficacy signal here. I can't remember the specifics. It has not yet been shown to have a definite, safe, and good efficacy effect yet in RP, which is what it was specifically being mentioned as a possibility for. And I do a good example here. I have patients with totally other inherited retinal dystrophies that even in my achromatopsia patients or XLRS patients, where it probably hasn't been specifically looked at. So not all these drugs are panaceas. It might be, if it is going to be effective, it might be very effective. It might be effective for one specific genotype, and it really should be looked at in a clinical trial before we start just using it off-label. I'll give another example of that, which is valproic acid. So, and this is actually a good historical discussion with what FFB and the role that FFB has played in examples like this. So valproic acid is an FDA-approved therapy for another condition. You can get valproic acid, but it was not approved yet for RP. And so Shailesh Kaushal and some investigators, actually from Florida, had some preliminary research and preclinical data that it might be useful, and they started distributing it and using it off-label in patients. And to the point where there's enough of a question out there that maybe does this work? And so the FFB said, well, let's really look at it. If we're going to start telling patients to take it, we need to have some safety and efficacy data, so let's do a little trial. So they did a 1-year trial recently. Todd can tell us the dates of it, on the valproic acid. I know Mark Panisi was a really active site. And they treated patients with valproic acid with RP, and then they looked at them for a year, and they followed safety. And it ended up not panning out, so it didn't show the efficacy signal that had been hoped for, desired, and that has sort of solved the problem of people using off-label valproic acid, which is, it can be, if it worked, that'd be great. It'd be worth the safety risk, but it does have risks. It can be liver toxic, and actually, unfortunately, some patients who have taken valproic acid have died from liver disease. It can cause really aggressive liver toxicity and damage. So, anti-abuse, if you were to take anti-abuse, you could never have a sip of alcohol or you would vomit. So that's how that drug works. It was to help alcoholics get off of alcohol. So there certainly are side effects, which may or may not be an issue for you and your family, but that also means that blood work needs to be followed. That drug is doing something. So it has a signal that needs to be followed, and we just don't have data. So I would always say, me being the investigator person here, that we need to study it in the clinical trial first before we just start using it willy-nilly and have potential safety signals.
Good morning.
Yeah,
This
Todd,
is
let me just say that Todd just brought up there are a number of interactions that drug has, and that is true with other medications you might be on. So never, ever, ever take something, even a vitamin, without discussing it with your primary care. And N-acetylcysteine is another antioxidant, and we can talk about that in a different day, but that also needs to be discussed with your primary and has interactions with other drugs. Sorry, where was the question?
Good morning. This is Dan Day. I'm a volunteer here in Orlando. Thanks for the presentation today. I have been told that if you participate in a clinical trial, you should assume that it's quite possible, maybe even probable, that you would probably be ineligible for other trials, although you would certainly not be ineligible for the treatment if it becomes FDA approved, but you probably should assume that you probably wouldn't be eligible for future trials of other things. Could you clarify that one a bit, please?
It depends on the previous therapy. So if you were in a previous natural history trial, you're good to go with anything. No one's going to care about that. If you were in a previous oral drug trial, for instance, the drug Alkeus, the company Alkeus Pharmaceuticals, I'm an investigator for that company. They have an oral drug that's a modified or deuterated form of vitamin A for Stargardt disease, Acusella. Mixostat. So there's these oral drug trials. A lot of times, if you've been in a trial that just uses an oral drug, you could potentially, and it's not a guarantee, but you could potentially be in another interventional trial if you, and the protocols will write in a washout period. So for instance, I was also an investigator for Iveric Bio. They were doing an intravitreal injection for Stargardt disease with a complement-related inhibitor pathway drug. And in the Iveric Bio protocol, you were allowed to recruit patients who had been in prior oral drug trials if they had a 3-month washout period. So that's just an example. Same thing when I look at my gene therapy protocols, you can never have had a previous invasive either injection or subretinal, certainly not a surgery with a gene therapy product. That will be a clear exclusion to really any other interventional trial. So just basically, if you've had something invasive done, so a shot in the eye or a surgery to the eye, then likely you will not be candidate for another interventional trial because those will specifically be listed as an exclusion. But you're right, you might still be able to have the drug commercially, although there are also immune-related issues with repeat gene therapy that we would need to discuss and think about, peculiar to the treatment that you're talking about. But again, it just depends on the therapy, depends on the mode of delivery, to answer your question.
Hi, I'm Brandy Moody. We're from Houston. I've got a 14-year-old son who has X-linked RP, and we haven't, done any trials or anything like that. And he's very stable. Like his disease hasn't progressed in really over five, maybe 6 years. So it's just nerve-wracking to try to figure out, do we just wait on something to be approved, or since his disease is stable right now, and we're also about to switch doctors, so I know we'll get a lot of advice from the new doctor. But what would you say to that?
Sorry, how old did you say he is?
14.
Okay. So, and I don't need to ask specifically. A couple questions there would be, who is the doctor that he's seeing, and what tests are they doing to establish stability? So we do like to follow ideally if you can be seeing an IRD specialist, and having, the testing that we would look at, for instance, in a natural history trial, the critical tests that we're following are the visual field, and that might be static perimetry or microperimetry, and then the OCT to follow that ellipsoid zone. A patient may be symptomatically stable, but might still be having some progression and loss of structure that we need to at least follow. And it's not to say you should go rush into a trial, and it's great that he's symptomatically stable. I'm not minimizing that. That's very important. But I think in addition to following the symptoms, we need to follow the structure and the visual function by the outcome measures to really watch, are those photoreceptors doing okay? Are we losing some ellipsoid zone, which is the foveal signal for photoreceptors, the visual field, the microperimetry. There always is that natural history trial, so you could kind of delay some time and be involved in a natural history trial. And then I would, again, think about the different trials going on and talk to the site. So if you have a specific question, I always say, directly refer you to that particular trial and pick a site. You don't necessarily want to talk to someone within the company. They're going to refer you to a site anyway. But so if you can contact, if you go to clinicaltrials.gov and think about those trials you're interested in the MIR, GTX, AGTC, et cetera, it'll have the contact listed for the PIs, whoever site's closest to you, and then you want to directly reach out to their clinical coordinator and ask these questions, because I think it really should be answered, and once they've seen your medical records and his medical records, if he's a good candidate.
Okay, thank you. And one last question is, he did have gene testing, but it's been many years since, I think it was 8. Is it good to have the testing done again, since tests have changed and been updated and things?
If he has a confirmed CLIA certified result that's a pathogenic mutation in RPGR, he's good to go. They might actually repeat it in the clinical trial. Sometimes they'll have in the protocol that it has to be Blueprint Genetics results, so just to throw out a name. But if it's pathogenic and if it was a CLIA certified lab, there's nothing that we're going to find. RPGR is a hard gene to sequence, so just make sure that is indeed the case. And then it's not RP2, which is the other cause of X-linked RP, so just...
Yeah, he's got RPGR, is what this says.
Good.
Yeah.
Yeah.
Okay, thank you.
I'll ask another question. Maybe both of the speakers can address this one. Some of these clinical trials take a year, sometimes 2 years. How do investigators know about emerging safety signals? So you're recruiting a potential patient to participate. Dr. K, how do you stay on top of the emerging information? And and maybe Rob, you can address the question, what's the sponsor's obligations?
start with the investigator side. It's interesting. It reminds me of what we all very sadly learned from again, nationally and internationally from the Jesse Elzinga case, which was, I'm sure you all historically remember, one of the things that really rocked the world of gene therapy was the unfortunate death of a young patient who was enrolled in a systemic, let's all remember this, it was intravenous systemic gene therapy, and he had a profound, robust inflammatory allergic reaction. He died a few days later. And so lawsuits and all kinds of things happened after that, and it really kind of rocked the world and slowed down gene therapy for years. Luckily, we've moved on and learned a lot of things. But one of the things we learned was at the time they weren't necessarily doing things like updates to informed consents and updates and amendments to investigator brochures when big safety signals arose in a trial. And if you look at the history and read about that on Google, you learn that there were actually some previous patients that had prior to him being treated that had pretty severe inflammatory reactions, and some of the preclinical data and the NHP monkey had some inflammation, and it wasn't at the time necessarily distributed as it should have been or as it is now to the patient knowing that. And so it's all about transparency and then up-to-date communication and knowledge of what's going on. So within these clinical trials, and the trials that I'm involved in are usually pretty small, I would say maximum 10 sites, some of them I'd say maybe 3 to 4 sites within the US, some of them 2. And so we all talk, all the investigators, we have frequent investigator meetings, and then the Data Safety Monitoring Committee has within the protocol a certain number of times they have to look at data, usually in between each dose escalation cohort. If something serious happens, which we call a serious adverse event or an SAE, we also use the term serious unexpected SUSAR, which I never, what's the S? Serious, unexpected, suspected adverse reaction. I don't know who made up that term, but SUSAR, and then you actually get a mailing from the sponsor, like say it happens in Europe and it's some other site that I wouldn't be talking to. We have to sign it that day, and I always get this 20-page thing I have to sign every single page and date it, that some site in Germany had a SUSAR on one of their patients, and so the sponsor distributes that to the sites, especially for international trials where you might not be hearing about these things. And so the investigator is updated of serious adverse and SUSARs, which are drug-related. And then what do we do? So you don't just immediately call every patient on the phone and tell them this, but when they come through your clinic, if it's enough of a change of a safety event that they need to talk to the FDA, the sponsor actually sometimes has enough of a safety issue that they have to talk to the FDA. They sometimes have enough of an issue that they need to update the informed consent, and then the investigator brochure once a year gets updated with all of these events. So the investigators are being updated with these things. But you're right, in real time, we have to really stay on top of things, and sometimes we have these trials where patients are treated 1 week apart, even protocols that allow patients at one site to be treated one day and the next day another patient to be treated, depends on how the protocol is written. So I have to stay in good communication with my collaborators, and we have to talk with each other if we're seeing things to be in real time, because obviously FDA and sponsors and updating investigator brochures is a little slower than me calling Mark Panisi and saying, what did you see in the patient you treated last week? So a lot of it is open lines of communication with the sites and the investigators, open lines of communication with your sponsor. If I see anything that I'm concerned about, I immediately call, and I know within each company who the person is I need, my point person. It might be a medical monitor, it might be the CMO, it's somebody in the company that I know knows what I'm going to be, knows the answer to my question. I call that person and say, look, I just injected this eye and I'm seeing some inflammation. Let's get the ball rolling for an investigator meeting. I've even flagged myself internally, said I want a DSMC ad hoc meeting to discuss this, and then we have a DSMC ad hoc meeting that I present the data. Ad hoc means it's not scheduled, it's added on because something popped up. So we're trying to stay on top of all of these things and update the patients and families in real time.
Okay, I'm Bob Watkins from Midland, Texas. I serve on Richard Fabian's Visionary Circle, and we went to our first convention, Foundation Fighting Convention, back in 2015, and at that time there were a number of foreign countries. There are over 20 or so involved with people that were attending that conference, and then we went shortly after that, 4 or 5 years ago, I don't remember the exact date, we were invited to go to the one at UCLA sponsored by France, and they had a number of speakers there from France and other countries, Germany in particular, And I noticed at this conference there's been no mention of what our cooperation, what Foundation Fighting Blindness cooperation is with the foreign countries, or why is there no other country here besides U.S. at this conference? Are we still connected with overseas?
No, not really. I'm not sure what, I don't know if you.
Yeah, I think your question is about the foundation's relationship with other organizations.
Right, with other countries and their trials and so forth, and what progress. That was what the one at UCLA was about.
Yeah, so this, we do have professional relationships with other similar organizations in Europe. The ERNI, I think, is one. We have relationships with Retina International, Fighting Blindness Canada. My understanding, though, this particular conference is for affected individuals and their families, primarily here in the U.S. But we do have other conferences where we do collaborate with international organizations.
Okay, very good.
Well, I would like to thank everyone for attending this session and recognize there's one more question back there. Okay, we have a question.
This is just a short question. 20 years ago, I was in the vitamin A palmitate study, but my defective gene has never been discovered yet, so you probably have to know that before you're in some of these new studies, don't you?
Sorry, what did you say your result was 20 years ago? I couldn't hear that.
I was in the vitamin A palmitate.
Oh, you're in vitamin A studies. Okay.
Yeah, but I still don't.
You don't know your gene.
They don't know my defective gene, and you probably have to know that before you're in any other study, wouldn't you?
No. So number one, if you haven't had recent genetic testing within the last 3 years, the panels have gotten better and wider, and I would definitely recommend a good panel like the Blueprint Genetics panels, who we use for the Foundation Fighting Blindness, My Retina Tracker genetic testing, so repeat genetic testing to see if we can find your mutation, just because it's important to know. But I always tell patients, even with that, the likelihood that this particular result for RP is going to get you into a potential clinical trial is still low, and the reason is there's the RPGR trials that we discussed. There's a few other disease, gene therapy specific related disease trials for RP, but there are multiple, at least there are several, non-genotype specific therapies as well. So it is absolutely possible that you would be a candidate. For example, I've talked about this trial multiple times today, but the N-acetylcysteine trial, which it's called the NAC or NAC attack trial, is starting very soon. It's going to be a phase 3. It's Peter Campochero's group that did a phase 1/2 looking at N-acetylcysteine, which is a super strong antioxidant. It's actually FDA approved for Tylenol overdose, not FDA approved yet, of course, for RP. So this is a clinical trial to see if, just like valproic acid, should we use this drug for RP? Is it working? And so in that, the phase 1/2 showed preservation of visual field in the treated patients versus the untreated patients. It's not genotype specific because it's an antioxidant, so it sort of is that panacea idea of it would probably protect and help multiple different genotypes associated with RP. Then there are, of course, depending on your level of vision loss, optogenetics, which is not genotype specific, and that would be for patients who have low vision with RP, maybe hand motion or worse, and those therapies in patients with LP or worse have, there's the GenSight trial, and the nanoscope trial for optogenetics. So with patients with hand motion or light perception or worse who were treated, they've had some improvement, even to count fingers in some patients. But as you can see, it wouldn't necessarily be a benefit if you were 20/40 vision, because it doesn't have that capacity and resolution to improve vision. But that's the idea of re-engineering the remaining healthy inner retinal cells to become photosensitive. And again, it's not genotype specific. And then there are drug devices, Argus, which is no longer being used, but that was not genotype specific. And then there are neurotrophic factor studies and antioxidant studies, oral drug studies. So there are multiple other potential trials. It just really does depend on your diagnosis. Your genotype is still important, and then work with your investigator to discuss what possible trials you would be a candidate for.
Okay, thank you. That concludes the session this morning. I want to thank our speakers.